welcome again to our video series where we interview scientists doing innovative research. Today, I'm really excited to have two scientists from the University of California, San Diego, my alma mater, and with some really great research that they are going to talk about. We have Dr. Pradipta Ghosh, who is a professor at the Department of Medicine and Cellular and Molecular Medicine at the University of California. She's also the director at the Institute for Network Medicine and the executive director and co-founder of the Humanoid Center of Research and Excellence. We also have with us Courtney Tindle, who is the program manager and director at the Humanoid Center of Research Excellence and the project lead for Crohn's disease. So welcome both of you.
Thank you, Annabelle.
Thank you.
Okay.
Thank you, Annabelle. Thank you both so much again for being here. We really appreciate it. This came together on very short notice because Annabelle and I were very excited when we read your preprint paper, which we'd like to talk more about today. But before we get into that paper, could you just briefly introduce yourselves and tell us why this particular research? What made you interested in this area of research? And I know your research interest may be broader as well and may involve other areas, but what was it in particular that led you down the path of researching Crohn's disease? So Dr. Ghosh, maybe we'll start with you.
Sure, thank you, Ildiko. So, by training, I'm a cell biologist. And one of the most fascinating thing, at least in my world, is to figure out how cells talk within itself, outside with things on the outside, whether it is bugs, toxins, things like that. And I try to look at it as whatever it is that a disease state has is somewhat of a broken communication, cell talks in those situations. So, and my second part of what I do is I'm actually a trained gastroenterologist.
Oh.
So I practiced, I was trained here at UC San Diego, did my GI and hepatology fellowship, and I took care of patients up until 2021 April, and then research took over from 90% to 100% of my life. And it just felt more rewarding to me that maybe some of the things that we could do might have a way for me to reach out to patients in a different way, perhaps in a more impactful way, perhaps more patients, if I just stuck to research and allowed my colleagues to save lives on the forefront, So, so having said that, how did I get into inflammatory bowel disease research is, we wanted to look at immune cell as just another cell, and our innate immune system, which is the epithelial lining of the gut, how it looks at the bugs inside, the immune cells right underneath that epithelium that is also looking at bugs that leak through the barrier. And we were simply asking the question, how does this lining of the gut that we have, how does it protect us in health? And then how does it go completely rogue in these very chronic disease states, like IBD being the poster child of it? And the age group it impacts is just so critical that there should be better treatment strategies, because the young, healthy kids at the prime of their life being hit with a diagnosis that will stay lifelong. And so that's what inspired us to have a big disease focus within our program to go into a territory that is multifactorial, no good treatment, no good animal models, let's do it attitude. let's do it in the way that should matter, which is human disease in human model and knowing what drives those diseases using those models. So that was the inspiration. With that, I'll let Courtney elaborate.
Well, I had my training for my master's degree looking at gastrointestinal infections. So I taught for a little while, and then I saw that Humanoid had a position. I was lucky enough to get it, and the main focus of research at that was Crohn's disease, and ever since then I've been studying Crohn's disease. So it was something new to me, but it's been, I guess, a great adventure learning new things about the disease and building the model.
Thank you. That's great and very helpful. And I think actually a perfect segue from what both of you said, which we completely agree with all of that. You know, the diseases are certainly suboptimal. There's a huge unmet need. It's a very complex, heterogeneous disease. But before we dive into unpacking perhaps that heterogeneity, I want to talk a little bit about organoids, because in this paper, you've used an organoid model. And again, to your excellent point, you know, mice are not an optimal model. They're useful for some things, but not an optimal model for IBD. So using organoids seems to be becoming more common. Propel, through Annabelle's leadership, has actually funded a study at Stanford using organoids as well. But I'd like to understand better, and for everybody watching who's sort of wondering, what exactly is an organoid and how do you make it work? Could you just talk for a moment, either or both of you, about organoids?
Sure. How about I hand over after patient to you, Courtney, because
Okay.
that's how exactly it happens. You know, I want to take one step back here to acknowledge organoid research it's both hope and hype. We hope this is going to be the thing that'll get us past this translational gap where things work wonderfully in mice and we cure them of every ailment we know of, and yet nothing works in humans. But with that comes a little bit of skepticism that is extremely well deserved for this organoid field, which is, imagine if every lab in this world used cell lines, and you can buy the exact same cell line. However, organoids are so unique to the patient it comes from. It's also a very expensive technology, very early on, untested, not easy to reproduce because of IRB guidelines of sending a patient sample to another lab to test and reproduce data. So I must acknowledge upfront that these are some real world concerns, that valid criticisms that people have for this technology that we have also used here. And at this point, I must say that patients and their caregiver, first-line doctors who took care of these patients for this particular study, need the biggest shout out here. Because if they did not participate in the study, give a part of their colon, the doctors who worked tirelessly to get them enrolled in this study, it was a mammoth initiative. And also research like this cannot happen with little dollar here, a dollar there. We were so incredibly fortunate, so grateful to the Helmsley Foundation to be allowing us and giving us this mandate. Go figure it out. Find out if there's a phenotype that matched a genotype so that we know that this is a group of patients. There's so much heterogeneity. So with that in mind, the recruitment of patients happen in the IBD clinic that we are so fortunate to have here, one one in the nation of kind at UC San Diego, patient samples are then transferred from the IBD clinic directly to the Humanoid Center. Courtney, you take it away from there, telling how we make these little organoids right out of them.
Yes, so we'll get a small bite-sized piece of tissue and digest connective tissue. And there has to be stem cells present within that tissue piece. Once the digestion process is done, they're seated in like a protein scaffolding called matrigel. And with the appropriate medium, then they self-assemble into these ball-like organoid structures that should mimic what is actually in the patient that it was derived from. And that is what we have been seeing through our RNA-seq studies.
So it's essentially in a petri dish, correct?
So like a 24 well or a 12 well plate, each well has multiple organoids that are like little mini balls of intestine.
And the interesting thing here is for thinking about it with a big picture is this allows us to put a patient in a well. So it is almost like one, if they wanted to test something in them, you could have a whole plate with actually a living patient's part that can be expanded over time to do so many studies that we did in this particular paper, at least. The second part is we use adult stem cells. So just to back up and say what they are, these are stem cells that are in that gut. So they know what is it to be a gut. They know what is it to be in the diseased gut of that particular patient. And with all the bacteria and toxins and everything else that was there in that diseased gut. As opposed to what some researchers do, which we did not use here, is taking blood or skin cells and making them go become a stem cell, and then taking that stem cell and making it become a gut. Why did we not take that approach? Because research has currently, from multiple groups at least, have shown that doing that not only makes that stem cell remember that it came from a blood cell, remember that it came from a skin cell, it also will not know what was in the gut of that patient. What was it facing? What was the inflammation? What was the bacteria? It will have absolutely no idea of what is it about being in a gut. And we call this, we call it epigenetics. So we have genetics, but then we also have what we call as what are we exposed to all throughout the time. What our cell is exposed to is imprinted on its gene expression as epigenetics. And that is what we've tried to preserve in the organoids that were derived from the stem cells from the colon in that patient.
Yes, and we're jumping ahead in the paper a little bit, but I loved the term epigenetic memory because you're essentially describing, as you just said, the environment or whatever factors it was exposed to. So you're not actually taking the microbiome and putting it into your petri dish, at least not yet. Maybe that's something someone will do at some point. You're not necessarily taking those, toxins or food proteins or whatever else might have been exposed, but there is seemingly some kind of epigenetic memory in these adult stem cells that you're using. Is that the right characterization?
Yeah, actually, to be honest with you, we were so incredibly surprised that you take these cells away from the immune cell, away from the stromal cell, away from its trillions of microbes hitting it, and you're in this very sterile culture technique, and yet it remembered where it came from. And so I have this same, maybe somebody else very smart said it, but I kind of say it this way to people I train in life or for this organoids, is good to know, remember where you came from to know where you're headed. And we have no way of using these systems to study a disease if those systems don't remember where they came from. So memory was important for us to preserve. And one of the things we decided very early on is to use adult stem cells as opposed to the blood or skin derived stem cells.
Makes sense. I think it was Maya Angelou maybe who said something like that. So
it is possible.
Yes. So that's great. So, okay, so we have these organoids and we have this problem of the heterogeneity of the disease, and patients will know that when they're diagnosed with Crohn's disease, they're typically classified into some type of clinical phenotype. So most patients are diagnosed as a non-stricturing, non-penetrating type of disease. So basically inflammatory. Some patients are diagnosed as stricturing. Some patients are diagnosed as penetrating or perianal disease. And actually, and this is a question I want to get to later, so maybe to think about now, some of the patients of an inflammatory phenotype then progress. A non-insignificant number over time will progress to one of the other phenotypes as well. So I do have questions about what that means for the molecular subtype, but that's getting ahead of ourselves. So for now, what you've done is you've looked at those phenotypes and said, okay, but if we do if we look at the molecular characteristics, do we see these three? And that's not exactly what you found. So tell us a little bit about what you found.
Sure, I can start it off, Courtney, and then hand over somewhere in the middle. So the classification that you mentioned earlier, Ildiko, is what we refer to it as Montreal classification, is how we clinicians put patients in bins because they look different on the outside. And one of the most frustrating things in Crohn's disease has been that despite all of that effort in putting them into bins, like you said, this patient goes from this bin to this bin after a while, and then from this to this. So disease evolves. Things change, the cell learns and adapts and becomes somebody else over time. And so does the patient. And the second frustrating thing that has been around and widely recognized in Crohn's disease among clinicians is the incredible frustration of having genetic drivers. We know what some of the most important genes are, like NOD2, ATG16L1, which are all in microbe sensing and clearance pathway. And yet, genetics and the clinical subtypes have no meaning whatsoever. And that has been one of the problems, because you think that we should be able to classify things by genetics and go at drugs and perfectly matched therapeutics. Didn't happen. So with that said, what the molecular subtypes emerged was actually the way it happened, and something that we could never put it in the manuscript was, you do the study, you get a few patients first, and you do the RNA-seq, which is looking at the gene expression, and you throw them on a map in a three-dimensional space we call principal component analysis. And we see the healthy here, and we see the Crohn's disease suddenly become two different separate types. And we were happy, surprised, dug in, figured it out what those subtypes are telling us. One of the subtypes were, they don't have too much inflammation, and yet we were treating them with anti-inflammatory drugs. What they had was a dearth of inflammation. They did not know how to mount an immune response. Forget about having too much of it. Whereas the other subtype was interesting. It showed us aging cells. aging cells that get badly programmed, they have DNA damage accumulating, and they spit out cytokines that can make fibroblasts, which are the stromal cells, produce a lot of stricture, collagen. And so one side was a problem in dealing with infection because I don't have enough inflammation mounted in response to it. The other side was more of I'm aging and I'm fibrosing, scarring. But the funny part that I wanted to tell you is, well, we did some more patients after a while. And I distinctly remember this because it was 1 a.m. in the night, and I typically don't do that, but I had something going on that night. And the student, graduate student who was working, Shahar, who had first done that map, suddenly sends me this text at 1 a.m., you gotta see this. I said, what? Well, we sent the second batch of patients and they fell exactly in those two clusters. So we all crave for reproducibility. We all want to see it work each time, but I was not one of them who was that ambitious that you take a heterogeneous cohort of Crohn's disease and they fall again back in the two clusters. So you can imagine it was 1 a.m. and we are both delirious and texting each other. And that worked. It reproduced even within the study itself. And so I'll pass on to Courtney. Now, Courtney, between those two subtypes, what did the phenotype show? Because she's the one who led those studies. And I'll hand it over here.
Well, yeah, I want to start and kind of like back up a little bit because I work directly with the scientists doing the wet lab studies. And I think it was almost a year of doing experiments where we were trying to put all of our data in terms of the Montreal classifications. And we were constantly almost like pulling our hair out, like, this doesn't make sense. We're trying to make sense of what is the pattern? And we couldn't really find one. So the moment that we found out there was 2 subtype clusters, we started to like reshuffle all of our data and put them in these bins. Then all of a sudden we can see that our wet lab experiments were matching what the RNA-seq said. So which is the figures that continued in the paper. I'm not sure if we'll get to those. So, and then again, another interesting thing is we would have the clinician tell us, oh, this subject changed from non-stricturing, non-penetrating to penetrating. Then again, that in the Montreal classification, we're always switching around data points, you know, trying to make sense of the data. But when you have the 2 subtype clusters that after a second run, they just stayed in the same cluster, it makes you really kind of think, should we think about this differently?
Mm-hmm. And thank you for reminding me that. See, these things don't go into the paper. And that's not how we wrote the paper. In the paper, we wrote, well, we were smart enough to do the RNA-seq first, figure out the 2 subtypes, and we wrote it that way. Truth is, we weren't smart enough to figure it out because we thought there are 4 types of Crohn's. Let's figure out what the 4 types are. The answer was nothing matches those, like Courtney said. So that these are things that didn't make it into the paper as to how the real world confusion looks like. But as patients and mothers of patients, you know exactly that's how confusing this disease is.
Right.
It's extraordinary, and it makes sense. You know, often the best discoveries are a little bit of trial and error and you know, seeing things, but you were also I think all smart enough to see the importance of that observation that you were making about these 2 clusters, and then asking the next important follow-up question is this reproducible? Is this really something that holds? And I think this is so important because, again, as we talked a little bit about before going on camera, you know, what is so challenging about Crohn's is that we don't know which patient is going to respond to which medication for how long. We know that the remission rates are still quite low. They're well below 50%, typically, especially over time. And so having a better understanding of truly the drivers of the disease, the two different phenotypes of disease, if there are in fact two, but that's so important. And you did some amazing work as well, looking at therapies, which we'll get to in a minute. But the idea of just having a better classification, I think, is so critical. And I've heard a lot of, very brilliant GIs talk about how we need to revisit that Montreal classification even before this paper. But I think this is one of the papers that is going to help, hopefully do that very thing. So I think it's amazing what you guys found. So basically you found two distinct subtypes, and like you said, you plot them and they're kind of at opposite ends, and you have healthy people here and you have these two subtypes over here. And so the first one, which you talked about a little bit, I find very interesting. It's basically an immune deficiency, is what you're saying. There's not the cytokine response that we think. It's not an overreaction of the immune system. It's actually a failure to clear microbes. So you called that?
Immune deficient infectious Crohn's disease.
Perfect. And then the second type, explain the second type with the, and you did as well, but to do with the aging of the cells and,
yes.
and oxidative stress. Explain that phenotype a little bit more, because I think the first one, at least for me, is easier to wrap my head around. The second one is maybe a little bit more difficult.
Sure. So I'll quickly revisit the first one to
Sure.
just finish off one thing. Anytime a cell is infected, it has to have some immune response, which is called our immunity, but not overzealous response, which will cause collateral damage. And what we found is, although it's widely believed that IBD is overzealous response, not true. Turns out it is less than what we even need to get rid of the bacteria, digest it, and clear up the infection. And that is the phenomenon that was, some people suspected that Crohn's disease could be this, that it is an inability, it could be a paradoxical state of immune suppression causing smoldering infection and hence smoldering inflammation. So that is one type. The second type, it's what these epithelial cells, if they, anywhere in our body actually, it is known that if they undergo some oxidative stress can cause RNA and DNA to get oxidized. They drive a lot of mutation accumulation over time. They assume what we call what an aged cell does with aging, cellular aging. And in that state, as if a cell does not die, it could enter a very problematic state that we call SASP, senescence associated secretory phenotype. What SASP means is it's an aged cell that spews out lots and lots of interesting cytokines that are programmed to drive fibrosis. It makes the surrounding cells react and produce a lot of scar tissue. As you know, a significant proportion of Crohn's disease patients have the complication of fibrotic tracts, fistulas, or even fibrotic stenosis of the lumen, causing intestinal blockade, requiring multiple surgeries. Those patients perhaps tend to have more of, the fibrostenotic disease patients had both kinds of infectious immune deficient state, as well as some of them were in the senescent associated fibrotic phenotype state. What we did see that the second molecular subtype, basically has a problem with stuck in an age state, producing cytokines with that underlying DNA damage. They are malprogrammed. They produce the cytokines to drive fibrosis around them. So that is our second molecular subtype. Courtney has the, we can briefly say the experiments that showed these two phenotypes, if you'd like. Courtney, do you want to?
Yes, so we did some bacterial clearance assays, and we would see that some had issues clearing the bugs, which were under the gray cluster, the immunodeficient cluster. And then we saw, for example, H&E, which is, we put our organoids into slides that were hematoxylin stained, and we would see that some of them had nuclear, that their nucleus kind of like blood bubbling, so nuclear atopia, which we found that were mostly in that yellow cluster that had the oxidative stress cellular senescence cluster.
Yes, and it's interesting, going back to your infectious disease molecular subtype, one of the, or maybe only, but one of the bacteria you used was the AIEC, the E. coli, that we've done a previous video about that. We talked to Dr. Brian Coombs, who's done a lot of amazing research with that and its relevance in Crohn's. So you used that, derived it, I believe, from a Crohn's patient, and then put it into the organoid and saw that there was a slow or imperfect clearance, which I just thought was so fascinating.
Oh, Courtney's handwork. I
Yes.
cannot take credit. But yes, we didn't derive the bacteria. We simply stood on the shoulders of giants that you have interviewed before who have contributed to this field's growth. And we were happy that we could use that as a tool, then go with stool slurry, which might make interpretation very difficult as a first study. And we hope to go into stool studies after that instead of using one bacteria. So we do acknowledge its oversimplification.
Sure, but it was interesting. And maybe your infectious disease background, Courtney, helped with that. But I think that, you know, I say this a lot. Sometimes I say it on Twitter, where I'm occasionally active in talking about these issues. And, you know, a lot of parents and patients will tell you that they feel that, like, intuitively, like there's some sort of underlying infection, like there's some, you know, and it's not universal. It's not everybody saying that, but you get a lot of people who have that intuition. And I wonder whether, you know, we need people like you with an infectious disease background, I think, to draw some of those parallels, to think of some of these experiments that you've done, and that others have done as well, to really try to see if there's something there or not. And I'll share one other thing, which our last interview was with Dr. Kate Jeffrey, who's at Moderna, and she's done some really amazing work in two areas that are sort of relevant to what you're doing as well. One is epigenetics, and finding an epigenetic signature in a small but relevant percentage of Crohn's patients, which I think is very interesting. And the second is finding an enterovirus B present in every resection sample that they looked at. It's not a huge number of samples, but it's not I can't remember the exact number. It's not a very small number either. And it was present in all of them. And so she's done some work, you know, characterizing that aspect. So again, you know, whether it's a bacteria or a virus or, you know, something else, the importance of trying to uncover that, I think, for patients and parents is so critical, because that's how you eventually get to targeting the underlying cause of disease. So I think your work in this area is so important. And then with the other phenotype or molecular subtype, I should say, also really critical, because those things won't work. You know, what you need to treat perhaps an underlying infection or you know, greater clearance of a pathogen is clearly not going to help that subtype. And so identifying these two, I think, is just amazing. Anyway, sorry for that little speech, but I thought it was great. So my next question.
I don't apologize, because you telling us what you read and you understood it makes us happy that we wrote it with enough clarity. So it's making us extremely happy that that is accessible, because that's the purpose of science, that it should be accessible. So
yes, thank you. And the preprint is great for that too. I mean, I assume you're going to hopefully publish this in a peer-reviewed journal as well.
yes.
So I hope a great one, high impact journal. That'll be great to read as well. But it's always nice when it's open access for us too. So the preprint is good from that perspective. In terms of understanding it, I think Annabelle and I are at the edge of our knowledge and capability, but I think we get the gist of it, and I think the gist is incredibly important. So thank you again for that.
I have a question. So between the two types, are there genetic, or did you do a genetic analysis as well? So I can't remember in the paper what came from that.
Yes, Courtney, all yours.
Yeah, so we took we went through like the catalog of IBD SNPs that are recorded in many papers, and kind of went through what are the main ones we should look at. For example, the gray cluster, we looked at NOD2 in the ATG SNP that we knew that is common in Crohn's disease, and we saw that when counting up the total SNPs that are between yellow and gray cluster, we saw that gray cluster was significantly more increased in those SNPs. Then same thing with the yellow cluster. We looked at some DNA damage SNPs, and we saw that the yellow cluster was significantly increased in the DNA damage. So we saw that there was matching between the genotype and the phenotype of the organoids that we had cultured.
And maybe Courtney's trying to be modest there, because I don't want to say we just cherry picked a few SNPs. What we did is we created the first ever 154,000 chip array, custom made for all IBD associated known genetics. Whatever SNP, anybody, any study, European, American, whatever cohort that was used. This would be the first comprehensive chip that combines genotype, a decent depth of multiple phenotypes being checked, transcriptome of those. And then one of the things that I didn't say it yet when we were talking on the organoid segment, I should make a small mention of it. Anybody can grow organoids because of the wonderful thing that Dr. Cleavers has given to the rest of the scientific community by showing that a stem cell can be made into an organoid. A lot of labs are growing it. How do we know that while growing them, we're not taking them farther and farther away from the truth? Because if we did that, we will be again fooling ourselves for another 10, 20 years, struggling to get anything out of these. So one of the things that Humanoid does is put ourselves through fire by taking organoid, healthy or disease, and actually taking the patient's epithelium, healthy or disease, and looking at them not by pathways, processes, and some gene enrichment analysis or any of those, but actually the exact match of gene to gene. This is a computational approach that no other organoid study that we came up with this actually as our benchmarking tool. Because for us, one of the things we prioritized was we don't want a model that's a lookalike. We don't want a model that is maybe lookalike. We want a model that is the closest replica to the real thing that is possible. And so this was one of the things that we did before even doing anything with the organoids. So that's something I thought I should mention.
And if I can just back up also to the 150K SNPs that were looked at, so this was a group effort of a lot of the co-authors sifting through papers, sifting through databases. And I mean, I think there maybe is even more that can be found in all that genotype information, but we were trying to make connections with our clusters and the genotype.
Right. And so you validated using a publicly available data set, right? For other, sorry, go ahead.
Yeah, we actually took multiple labs, Crohn's disease organoids, IBD organoids. Luckily, we're so grateful to these groups of scientists and all these journals who want us to put everything out there, because then we all can look at each other's things and compare. So we took everybody's data sets that is out there from organoid models, and we compared them all with the same measuring yardstick of actual patients lining from the gut with nothing done to it after that, meaning not cultured or anything. It's the exact lining from the colon. And when we did that, we were surprised that there are many organoids that have nothing left in them that looks like the actual disease. And there were some who did manage to keep it, not us just. So we found both kinds, and that made us have both hope and worry. We were hopeful that having standardized methods to grow these organoids in a type of condition that keeps the epigenetic memory can be defined. So there is hope. Worry, because we could see that prior publications with several of these organoids are probably, may not reflect the truth because they did not look like the actual disease. So with that, I think we were just trying to make sure there's a benchmark, and we wanted to make sure to be the ones to establish one if we could.
Yeah, and again, as organoids become more utilized, and I take your point that it's both hope and hype. I feel that way about many things, microbiome research, many things. So I think that's a really, really important point to make. But, just like with microbiome, it's that standardization and getting to the truth, as you say, really is the key. So anything that moves that knowledge and gives us more confidence in these models, I think is incredibly valuable to the field. So I appreciate the work done on that part as well. My next question is going to the therapies now, because you didn't just sort of stop there. You could have stopped and said, we identified these two different molecular subtypes, but you did something else, which at least from a parent and patient perspective, I think is, we're always looking for hope. At the end of the day, that's what people want, is to know that these diseases, they're incurable today, but that's just because we haven't cured them yet. And so what you did is you went a step further and you said, what if we try to apply therapies that are specific to each type of molecular subtype? So tell us a little bit about what you did there, Courtney, what experiments you did there, and then what you all found.
Yeah. So first we had to have a measure of senescence for the yellow cluster. So we used a senescence associated beta gal. And we saw that the yellow cluster had an increase in that assay, and we tested different therapeutics, and we saw that especially JAK1 and 2 inhibitors were important for reducing the senescence. And we saw that that was specific to yellow cluster and not to gray cluster.
And just to interrupt you, so JAK inhibitors are used, right, in IBD, and we know they're useful in IBD. So that's a really interesting finding that is, giving us more insight maybe into which patients can use those therapies. Although I'm not sure, are they JAK1 and 2 inhibitors on the market?
So I can help out here because it is incredibly frustrating when a phase 3 clinical trial goes belly up, because Courtney and I often discuss this, because that's a $800 million loss for our society each time that happens. And yet for IBD, that has happened. It has repeatedly happened. So what is interesting, since you brought the JAK inhibitor up that Courtney was just narrating, that not only did she show that the JAK inhibitor works to reverse senescence in the molecular subtype that had the problem of aging and all of this damaged DNA-related response, she could also show it is totally worthless in the other molecular subtype, the one that had difficulty in clearing the bugs. So imagine now, phase 3 clinical trial, we found about 70% of the patients are on the infectious immune suppression subtype. I do a phase 3 study. I don't know, but maybe about 70% of my cohort was of that subtype, but I gave them all JAK inhibitors. What happened? My phase 3 failed. So what happened is tofacitinib is not approved for Crohn's disease. It did make it for ulcerative colitis. Tofacitinib not only just hits all 3 JAKs, JAK1, JAK2, JAK3, there are 3 of them. 3 is not so important for senescence. For senescence, 1 and 2 are important. So Courtney was using one JAK1 specific on JAK1 2 inhibitors, not 3. So what happens with tofacitinib? It hits all 3 JAKs, lot more side effect than I need on a patient. And I have a trial done with 70% of the patients who should not have received it. So now if we take a step back, why did JAK inhibitors fail? Should the study be done differently? Should the enrollment have included only the patients who are likely to respond? So on and so forth. What about the infectious disease subtype, Courtney?
Yeah, so we saw that the PPAR alpha gamma dual agonist worked specifically for the gray cluster. And then the JAK inhibitors did not clear the bugs the same way. So we see that these therapeutics were specific for the subtype. And also
probiotics as well, probiotics and postbiotics. And
yes, the probiotics we saw more was helping with a tear deficiency. So the barrier was leaky.
Right.
And it helped with the leaky barrier and recovering from that. But that was mainly seen in the non-stricturing, non-penetrating that we saw had more of that leaky barrier phenotype.
And going back to the phenotype with the JAKs, and of course we have tofacitinib, and we now have upadacitinib as well, which it's interesting. I mean, again, maybe these are more effective in UC, but that one seems to have a fairly high remission rate. I wonder sometimes what the difference is between those two, but that's for another day. But in that molecular subtype where you use the JAK, You used a couple of other therapies as well, right? I can't remember off the top of my head what they were, but you tried a few different things and found some efficacy. So I almost wonder whether the reason I say it is because some of these, may have fewer side effects or risks associated with them. You could possibly use multiple of these agents together to get a more effective, theoretically. You'd have to study it, obviously, but there are different ways to target these same patterns that you found from a molecular standpoint, just based on the fact that you use different therapies for each of these underlying subtypes. I thought that was interesting.
Yeah, so for cellular senescence, we did use card-carrying senescence inhibitors. Some of them will never perhaps become a widely used drug because they are plant derivatives. Their pharmaceutical industry has very little appetite for things they cannot commercialize that way. So, but you're right, there are alternative medicines that can emerge out of this. Combination treatments can emerge, but there are also a lot of drugs of the class of senolytics, which is to kill the senescent cells and leave only the young ones there. So there is a lot of deep pocket to look into to find new classes of drugs for just that subtype. And most importantly, no matter which drug is taken to trial, we should choose the right patient to fit the right drug. Right now, we are doing one size fits all, which you started off very appropriately at the top of the hour, that's just not right, and probably the reason why we fail so often in phase 3, phase 2.
Yes, couldn't agree more. Okay, so you use different treatments for the 2 different subtypes. You do find in your studies that they reverse that subtype, correct? So they normalize whatever biomarker you're looking at for each of those subtypes.
Yes. So in developing a treatment, we had to take a step back to see how would this ever be a point of care? How would this ever translate? If a molecular subtype were ever to be something that is important, what can it be? Well, it could be that we can detect it, and hence we know which patient has it. How about we can track it? So we know how to screen compounds to see if it goes down or goes up. And finally, if we reverse it, we can even perhaps look for a drug's IC50, EC50 calculations based on how much did we reverse it. So our goal of making this translation, the impact of translating any such discovery had to be, well, could it be a step that can be added on before in the preclinical phase before we even touch a patient a trial, is to have this kind of a cohort of patients with different molecular subtypes screened for this particular readout that is disease-defining readout. So the disease-defining readout for the infectious phenotype was give it infection, doesn't produce the cytokine, doesn't clear it. That was our phenotype we were tracking. For the senescent one, the disease-defining phenotype was aging, and we were simply tracking cellular aging. So if that were to ever translate, we were thinking that compounds can be screened for new drugs, new treatment modalities, combination treatment, like you said. And so those would be perhaps the ways how we can think of this translating.
Well, that you've answered my next question, which was going to be, you read my mind, what's the next step from here? And how do we translate it? I think you answered a lot of what that next step is, and hopefully it is something that gets used in pre-screens. And sometimes the hardest thing is taking knowledge and applying it into clinic, translating it. We're not quite at the point where we're going to grow organoids for every patient, right? So for many of the reasons that you that you identified, some of the challenges you identified at the top of the discussion. So I guess the question is, practically speaking, outside of clinical trials, do you see us ever getting to a point where we do have kind of an organoid for every patient to determine what the appropriate therapy is? You could even test the appropriate therapy in the dish and determine if it works for that patient. Do you ever see that as a practical possibility?
I can go, Courtney, if you.
What is your ideas of practicality? I would like to.
I think so. One of the things I've learned, never say no. And the second thing I learned is if I say no, I'll be proven wrong. So with that in mind, let me tell you where it is currently being used that an organoid determines therapy. It's there for cancer. We are doing it for pancreatic cancer and some of the most, there is none of the chemotherapy works, but at least they are beginning to see some work better than the others to give a few more months of life. Where we do not have anything like that are non-cancer diseases. In a world where MRI and other ways of classifying these patients and repeated endoscopies, so many, there is no problem in getting access. Think of that way, because we do endoscopies so often on these patients. So if the access is not a problem, the cost of generating the organoid isn't so bad if it's done properly to keep meaning in it, that is. I do envision that it will, if that is the way to get a molecular subtype classification.
I was just curious because my thoughts were, we used organoids to find out that there were these subtypes, but like when taking a biopsy and doing RNA-seq studies, could it just be from the tissue level, looking at more of the expression profiles that are from the epithelium, because that's what we studied.
More of a point of care test can be derived out of what we have.
The organoid would just be a little bit more lengthy, but I don't think impossible scenario.
Yeah, the only potential benefit is to sort of pre-screen the therapies and see what might work.
Right.
You're absolutely right that you could do a lot of the classification just from the biopsy sample. 7 years ago, that was one of my many very naive questions when I knew very, very little was, shouldn't we be doing like a deeper dive into these? Like we take these biopsy samples, don't do that much. Pathologist looks at it, but you don't really try to figure out what's going on. We just have these sort of observational hallmarks to classify it as Crohn's or UC. It seems like a missed opportunity. It always felt like a missed opportunity to me to not use those biopsy samples to really have a deeper understanding on a personalized level at what's going on in that individual. But we're still not there yet, but we're pushing, we're hoping, we're working on that. Annabelle, do you have any other questions? I think I'm sort of gone through a lot of mine. And
yeah, no, you as usual, you've done a great job, Ildiko, and I really appreciate this because it's so interesting and hopeful. And as a parent, of course, and I think there are lots of parents will be watching this, will be very impatient, will want to know, well, what can we do? What can we do to speed things up? Because we want something to relieve our kids. I mean, like my son has gone through, I've counted it up, he's done about 8 different meds right now. He's on his 9th med. So, it would be good. It would be good to have something where we can actually take a sample, see what kind of Crohn's he has. I mean, specifically, oh yes, this is the kind of medicine he needs. That might just reverse it entirely. What a dream. That would be fantastic. So
I do have one other question, actually, because you took, and I forgot to mention this, I meant to, you took samples from from the colon specifically, and you have a good rationale for saying most studies have looked at, you know, the small bowel or terminal ileum in Crohn's patients, and so you want to look at the colon. And you also did take some samples from UC patients to compare. I'm curious, and I don't recall exactly, what it looks like a spectrum, does it not, somewhere between the, you know, UC and some of the subtypes of Crohn's look like they might overlap? What did you see between UC and your colonic Crohn's samples?
Yeah, so I think you brought up a couple of very important points here that we didn't emphasize earlier. Colon is involved in the majority of the patients, but it's more importantly, it's also the place where it has the trillion microbe problem. And one of the things we now know is whether it is the consistency of the stool when by the time it reaches the colon or whatever else it is, that the contact time with the colonic epithelium, which is the lining of the gut, is really is now felt to be a disease driver from everything from dementia to metabolic syndrome to whatnot. So we were very focused on the lining that had the most promise and also the most involved organ in the disease. The second part I wanted to address was among UC and CD patients, one of the biggest differences we clinically see is in ulcerative colitis, the disease is so limited to just the lining area. However, in Crohn's, the inflammation doesn't stop at the lining. It just keeps to continue and goes through the wall of the bowel. So it's a little bit more malignant, a little bit more penetrates through tissues. It has this weird ability. Being aware of that clinically was important as we were analyzing the RNA-seq data. And what we saw is the difficulty in handling microbe and clearing it was a common underlying theme between both ulcerative colitis and the Crohn's disease colon epithelium. Whereas the aging cell, DNA damaged aging cell that is just spewing hatred of cytokine around to cause fibrosis and stricturing, it was unique to Crohn's. And these, the aged cell with this very weird, it has forgotten how to be an epithelium almost. That's how these aged cells behaves. That makes a whole lot of sense because those cells can move. They can move through and through the wall, create fistulas, they can create strictures. So it makes sense how clinically we just don't see those things in ulcerative colitis. We see a very localized thing, and ulcerative colitis is a disease of the colon. Very rarely it may get to the terminal ileum, right? So we were so fascinated to see that the colonic epithelium of the majority of the Crohn's disease patients was dysfunctional, whether or not they had a colonic involvement clinically. And that was another important but quite insightful finding that we plan on following on through other studies.
Exactly. That's very interesting as well. So I guess that'll be my last question to wrap up is, you know, where do you plan to take this, if at all? Courtney, maybe that's for you, a question, but what do you plan to do next in this area? What can we look forward to? And, you know, everybody watching this can hopefully support your efforts, which is great. And a shout out to the Helmsley Foundation, like you said at the beginning, for supporting this really, really important work.
Well, I think we have another chapter of Crohn's disease right now that we're currently trying to, hopefully by the end of the year, wrap up for modeling intestinal fibrosis in a dish. So that's one thing to look forward to. And I think Pradipta, maybe you can discuss a few sentences your next year chapter.
Yes, so we were deeply committed in immunology space to go after something that is extremely difficult as a problem. Guess what? IBD was that, if one had to pick one. And what do I mean by difficult? Difficult because too many cells are involved, too many pathways are involved, too many factors are a problem. We don't know anything about it. Nothing works in animal model. And oh, by the way, 90% of the drugs don't work. So with that in mind, we actually did what Annabelle, you mentioned, the artificial intelligence guided drug discovery program. We launched that at the very beginning when the Institute for Network Medicine was going after challenges to pick. Naturally, it is actually our most advanced program for therapeutics in our institute. So we have bilateral effort from computation side to go after identifying high value drug targets, modeling side to say, here is the best model. We're at this point to now go fuse them. We have found macrophage targeted therapeutics earlier for Crohn's disease. Not at all dysfunctional ulcerative colitis, unique problem in the macrophage, and we put a drug to it in one of the manuscripts earlier. First manuscript went after the barrier function, something that has never been yet addressed with a specific barrier targeted drug. We are going after disease molecular subtype therapeutics, and the next one we are very excited to launch is going after the largest target class that treats from glaucoma to asthma to neurobehavioral symptoms to everything else, but underutilized in IBD, which is the GPCRs. The GPCRs that are 40% of the drugs on the market, we don't use them for IBD. Well, why not? Well, because there's no good way to go through 1,000 GPCRs to come up with the best horses to bet on. Our artificial intelligence program allows us to do that by blurring out what might not be important and only focusing on what could be. And then we go with Humanoid Center and say, here are our best targets, here's the drug, here are the cell types we want to go at. Humanoid has patients, organoids patient, myofibroblasts patient, blood cells, in order for us to go multimodal, multicellular, target-specific engagement to go at drug discovery in style that way. So those are all the future things that we are incredibly excited to dive into.
That is really incredibly exciting. That is exactly what we need and want. It's to use the technology that we have, whether it's developing better organoid models or using AI. I was involved in my pharmaceutical career of using AI pretty early on, and at a time when it really wasn't that effective yet for drug discovery. We're trying to make it work. It really wasn't working. I think it's gotten so much better since that time. That was about a decade ago, maybe a little more. And so important, I think, that we use, AI for many aspects, drug discovery being such a almost obvious good application of that technology, I think, because like you said, screening, thousands, millions, whatever molecules to try to figure out what might work is a tough job. So I commend you for doing that. I think at the end of the day, research, to the extent that it can help patients, it is really, really just, I think, one of, the best things that you could be doing. And I really appreciate how you guys think about these problems and what you're doing to try to move us forward and get to better therapies and better outcomes for patients. So thank you again so much. Annabelle?
Yeah, I just wanted to say, you know, I keep thinking about what you were saying, how you, when you had started, you were thinking along the Montreal classifications, and then it just sort of, it went into a totally different thing, and that you were open to accepting that and embracing that. And to me, not being a scientist, but, you know, looking at scientists, having an idea of what it must be like in a lab, that to me is like just perfect science. I mean, that's what it is, right? You go where the science takes you, and it's not, you don't go in with preconceived notion and forcing it through, you know, through a hole that doesn't, you know, that's squared or whatever. So yeah, I really appreciate that. So.
Well, thank you. I just want to make sure I mention one of our colleagues you invited is not here today, Dr. Shumita Das. I should mention that organoids and stem cell biology and host and microbe interplay is really her focus of science, her flavor of science. I'm just a cell biologist, and I'm a physician, so I come from that side. And there is, we both co-founded the Humanoid Center, and the foundation, Halsey Foundation, was just kind enough to trust us to do something with what they gave us. So there is also a connection because Courtney trained as her master's student in the laboratory of Dr. Das. So I just wanted to make sure, it's unfortunate she cannot be here today, but wanted to make sure that credit went where it's deserved as an incredibly important member of the team, without which the study would not have happened.
And also, I did not perform all these experiments by myself. I performed experiments, but this amount of work in the matter of 2 or 3 years would not have been able to be done without all of the co-authors that we have listed on the paper. I was there the whole time to help organize the data and come up. We all thought together of how we could do this, but everybody had a major part in this study.
Thank you both for saying that. Of course, it's a team effort. We will link the paper or the preprint in the description below the YouTube video, so people can look at it directly and see all the authors, and you have some beautiful charts and pictures in it as well, I should say. So people should look at it so that they can, you know, go look at those, and when we're talking about the, you know, yellow and gray, they can kind of visualize what that means. So thank you both again so much for taking the time today and for all of your work. It is so appreciated. So thank you.
Thank you.
Thank you. Thank you both for doing this. Thank you.